A paper just dropped that might end the 100-year war against sugar. Not reduce sugar, not substitute sugar. End the entire problem. Since 1879, when a chemist accidentally discovered saccharin by not washing his hands before dinner, humanity has been searching for something that tastes like sugar but does not destroy our bodies. We have tried everything. Aspartame, stevia, monk fruit, sucralose. Every single one comes with a catch, a weird aftertaste, digestive problems, an inability to actually cook with it, or that unsettling feeling that you are eating something that is not quite food. What if I told you that a team at Tufts University just figured out how to produce a sugar that has 92% of the sweetness of regular sugar, 40% fewer calories, does not spike your blood sugar, does not cause cavities, and browns in your oven exactly like the real thing, and the secret ingredient came from slime mold? This is not a future promise. This is a paper published in Cell Reports Physical Science in December 2025. And the results are kind of ridiculous. The sugar is called Tagatose. You have probably never heard of it, and there is a good reason for that. Tagatose exists naturally, but in such tiny amounts that extracting it was never economically viable. You can find traces of it in heated dairy products. A little bit forms when you make yogurt or cheese. Some fruits have it: apples, pineapples, oranges. But we are talking less than 0.2% of the sugars in those foods. It might as well not exist. Previous attempts to manufacture Tagatose hit a wall. The traditional process started with galactose, a relatively expensive sugar, and could only convert about 40 to 77% of it. The economics never worked. You cannot replace table sugar with something that costs 5 times as much to produce. So what changed? The Tufts team found an enzyme in slime mold. Let that sink in for a moment. Slime mold. One of the strangest organisms on Earth. Not quite plant, not quite animal, not quite fungus. A single-celled creature that can somehow solve mazes and optimize transportation networks. And buried in its biology was the key to unlocking cheap sugar alternatives. The enzyme is called galactose 1-phosphate selective phosphatase. You do not need to remember that. What you need to understand is what it does. It allows the researchers to reverse a natural biological pathway. Normally, your body uses something called the Lellouar pathway to convert galactose into glucose. It is a one-way street that has been running in the same direction for billions of years. But with this slime mold enzyme spliced into E. coli bacteria, the researchers flipped the pathway backwards. Now they can start with cheap, abundant glucose and convert it into galactose, which then gets converted into Tagatose. The yield, 95%. Compare that to 40 to 77% with the old methods. This is not an incremental improvement. This is a fundamental breakthrough in production efficiency. But here is where it gets interesting for your actual life. Tagatose does not behave like other sugar substitutes in your body. When you eat regular sugar, your small intestine absorbs it completely and your blood glucose spikes. When you eat Tagatose, only part of it gets absorbed. The rest travels to your colon, where gut bacteria ferment it. Clinical studies show minimal increases in blood glucose or insulin after consumption. For diabetics, this could be genuinely life-changing. And your teeth. Regular sugar feeds the bacteria that cause cavities. Tagatose appears to do the opposite. It actually limits the growth of some of those harmful microbes. There is even evidence it might have probiotic effects, promoting healthier bacteria in both your mouth and your gut. Now here's the part that every other sugar substitute has failed at: cooking. Stevia cannot brown. Aspartame breaks down when heated. Most artificial sweeteners cannot provide bulk, that physical volume that sugar adds to recipes. They are too concentrated, too different in their chemical behavior. Tagatose browns like sugar. It caramelizes. It provides bulk. In taste tests, it closely matches the flavor and mouthfeel of conventional sugar. You can bake with it and nobody would know the difference. Think about what that means. For the first time, we might have something that actually works across every use case. Your morning coffee, your birthday cake, the cookies you stress bake at 2 in the morning, all of it with 60% fewer calories and no blood sugar roller coaster. Let me be clear about what this paper actually shows and what it does not show. This is a lab demonstration. The researchers engineered E. coli bacteria in controlled conditions and achieved 95% yields. That is extraordinary science. But scaling from a laboratory to industrial production is a different challenge entirely. Processes that work beautifully at small scales often hit unexpected problems when you try to produce millions of tons. There is also the question of public perception. When people hear that their sugar was made by bacteria, some will have concerns. Never mind that we already use bacteria to make insulin, cheese, yogurt, and countless other products. The word bacteria triggers something in people, and Tagatose is not 0 calorie. It still has about 1.5 calories per gram compared to sugar's 4. If you are counting every calorie, this is better, but not a free pass to eat unlimited sweets. Finally, like other sugar alcohols, large amounts of Tagatose might cause gastrointestinal effects in some people. Your gut bacteria fermenting it can produce gas and discomfort if you go overboard. But even with those caveats, the significance of this research is hard to overstate. The lead researcher, Nick Nair, said something that stuck with me. Quote, "The key innovation was finding the slime mold enzyme and splicing it into our production bacteria. That allowed us to reverse a natural biological pathway that metabolizes galactose to glucose and instead generate galactose from glucose. Tagatose and potentially other rare sugars can be synthesized from that point." End quote. That last part is crucial, potentially other rare sugars. This is not just about Tagatose. The same approach could unlock an entire family of rare sugars with different properties, different benefits, different applications. The researchers have essentially built a platform technology. Here is what fascinates me about this discovery. For more than a century, the best minds in food science and biochemistry have been trying to solve this problem. They tried artificial molecules. They tried plant extracts. They tried fermentation of various substrates. And the answer was sitting in slime mold the whole time. Evolution had already figured it out. We just needed to find it and borrow it. That is the pattern of so many breakthroughs. The solution exists somewhere in nature, optimized over billions of years, waiting for us to recognize it. CRISPR came from bacteria defending themselves against viruses. Velcro came from burrs sticking to a dog's fur. And now revolutionary sugar production might come from the biochemistry of an organism that most people would scrape off a log. The 100-year quest for the perfect sugar substitute might finally be over. Not because we invented something entirely new, but because we learned to read the instruction manual that nature already wrote. Whether this specific technology reaches your grocery store depends on economics, regulation, scaling challenges, and a dozen other factors I cannot predict. But the science works. The proof of concept exists, and the door is now open. What other solutions are hiding in organisms we have barely studied? What else has evolution already solved that we are still struggling with? Sometimes the most advanced technology is just learning to copy nature better. And nature has been doing chemistry a lot longer than we have. If you want to stay updated on more discoveries that might actually change your daily life, there is always something new to explore.